Okay, so welcome to this next video in which we are discussing the use of glucocorticoids in the treatment of asthma. Okay, so we're currently talking about the pathology of asthma. Okay, so we've had our primary exposure to the allergen, and we're now going to launch an adaptive immune response against it. Now, this is where people who suffer from allergic asthma differ from people who don't suffer from allergic asthma. People who don't suffer from allergic asthma and are not allergic to this specific allergen will not launch an adaptive immune response against it. Okay, so everything we've discussed now and up until now is what will happen in someone who is allergic to it. And it's not really understood what point is actually different in this pathway for people who are allergic and for people who aren't allergic. There are many different steps that could be different, okay, and I think we should discuss these as we go along. So one of them we've actually gone past so far, and it's this signal 1. Okay, so we had this antigen presenting cell which had a fragment of the allergen on its surface, okay. Now, generally it is believed that this must happen in everyone, okay, so we can't understand why some people's macrophages would present this allergen and some wouldn't, okay, so the general assumption is that this must happen in everyone, okay, but then in the people who are allergic, you must be able to find a naive CD4 positive T cell that has a T cell receptor which is complementary to fragments of this allergen, okay? Whereas in people who aren't allergic, potentially they don't have naive CD4 positive T cells which have T cell receptors which will bind to this allergen fragment. So this could be one of those stages which is different between allergic individuals and non-allergic individuals. Now, another step that's very mysterious is what's going to happen next. So remember I told you that in order to actually activate the naive CD4 positive T cell, signal 1 is not enough. You have to provide co-stimulatory molecules on your surface. Now, these co-stimulatory molecules are not just expressed on the surface of antigen-presenting cells willy-nilly, so you don't usually have these molecules on their surface. Instead, in order to get the expression, you have to activate what are known as PRRs, and PRRs stands for pattern recognition receptors. Now, antigen-presenting cells have a huge number of different pattern recognition receptors, and they all bind to molecules which are collectively known as PAMPs. Okay, so what is a PAMP? Well, a PAMP stands for a pathogen-associated molecular pattern. So where shall I write that? Oh, I'm running out of space. Drat. I'll put it up here. Okay, so pathogen-associated molecular pattern. Now, these are molecules that pathogens would make and potentially have on their surface, which no human cell in its right mind would ever make, okay? So these are a dead giveaway that you have an invading pathogenic cell within your body, basically. So pathogen-associated molecular patterns is the umbrella term for all molecules that pathogens would have that human cells would never ever have. Okay, so there are a huge number of different pathogen-associated molecular patterns, and the different pathogen-associated molecular patterns will have different receptors on the surface of the antigen-presenting cells, and all of these receptors are collectively grouped together as pattern recognition receptors. And I want to emphasize that it's not like you just have one pattern recognition receptor which recognizes all PAMPs. No, each PAMP will have its own pattern recognition receptor, and when that PAMP is present in the cytoplasm, sorry, not in the cytoplasm, in the interstitial fluid, it will bind to the pattern recognition receptor on the surface of the antigen-presenting cell and activate the expression of these co-stimulatory molecules. Now, one of these co-stimulatory molecules that you have to produce is CD40, which we'll colour in yellow here. Now, the other one can be one of two proteins, okay? So there's a little bit of scope for versatility here, okay? So you can either use B7.1, which is also called CD80, or you can use B7.2, which is also called CD86. Okay, so usually these molecules are only put on the surface if a pathogen really is invading the body, okay? And this is a problem. How on earth does the antigen-presenting cell get activated to produce these two co-stimulatory molecules? Because it's presenting an allergen fragment. This allergen fragment was not a pathogen. It wasn't associated with a pathogen, okay? So it didn't have any PAMPs to activate the APC's pattern recognition receptor. So where did the antigen-presenting cell that is going to trigger off this entire humoral adaptive immune response against this allergen fragment, where did it get these co-stimulatory molecules from? Well, it's a bit of a mystery. Okay, and this is one of the key things that people are looking at as to whether this is the difference between people who suffer from the allergic response to this allergen and people who don't. Okay, however, suggestions for how this could happen is that potentially you were already ill, potentially you were ill, so potentially you had a viral infection, maybe a cold or flu, and you just happen to breathe in the allergen at the same time. Then the antigen-presenting cell could, phagocytose the allergen, do everything we've discussed so far, and its pattern recognition receptors would be activated by PAMPs that were from the actual pathogen, and that would induce the expression of the co-stimulatory molecules. And if the two happened concurrently, then you'd get both of these signals. That's a suggestion for how it could happen. Okay, but the problem with that is you'd think that would happen in everyone, so it takes away the idea that this could be something that's different between the allergic individual and the non-allergic individual, and it replaces it just with, it's a case of luck then, as to whether you breathe in the allergen for the first time whilst you're ill or not. Okay, right, so then what happens is these co-stimulatory molecules are going to stimulate receptors on the surface of the naive CD4 positive T cell. So whatever the mechanism of these co-stimulatory molecules being induced, they are induced, and they are going to now activate the receptors on the surface of the naive CD4 positive T cell. And the receptors on the surface are CD40 ligand, or CD40L, is the molecule to which CD40 is going to bind, so let's have this in pink here, okay, and CD28 is the protein on the surface of the naive CD4 positive T cell that either B7.1 or B7.2 is going to bind to, and we'll have this in vivid purple. Okay, right, so when CD40 ligand and CD28 have both bound their respective ligands, okay, or their respective proteins on the surface of the antigen-presenting cell, they give signal 2 to the naive CD4 positive T cell. Okay, and once you've got signal 1 and signal 2, you will then undergo the next stages that are going to lead to the activation of this naive CD4 positive T cell. Okay, so let's discuss these now. Right, okay, so what's going to happen within this naive CD4 positive T cell next? Well, it's going to start producing two things. Okay, so let's put these here. So we're going to start producing two separate proteins. One of the proteins we're going to start producing is interleukin-2, IL-2. Another protein we're going to produce is the interleukin-2 receptor alpha component. Okay, so basically the interleukin-2 is going to be secreted from the naive CD4 positive T cell, whilst the interleukin-2 receptor alpha component is going to go up and be implanted into the membrane. Now this is an important point to understand. Okay, so let's say this is the phospholipid bilayer. Now, naive CD4 positive T cells, before they have undergone signal 1 and signal 2 of activation, okay, so when they were just sitting in the lymph node inactive, they only had two parts of the interleukin-2 receptor. They had the interleukin-2 receptor beta component, IL-2R beta, and sometimes I'll put a dash in between interleukin and the number, and sometimes I won't, okay? I hope that doesn't confuse you. And sometimes, and the other thing they have is interleukin-2 receptor gamma. Okay, so they have two of these components expressed constitutively. However, if you only have beta and gamma, you do not have a functional interleukin-2 receptor. Instead, you have to add on the third and final component, which is the interleukin-2 receptor alpha. So once you've received signal 1 and signal 2, you start producing the interleukin-2 receptor alpha subunit, and then you get a complete interleukin-2 receptor on the surface of the T cells which have undergone signal 1 and signal 2. Okay, so here is now our complete interleukin-2 receptor. Then the T cell, which has undergone signal 1 and signal 2, has also secreted the interleukin-2, and the interleukin-2 will now act on the interleukin-2 receptor on the T cell that just released the interleukin-2. So this is what's known as autocrine signaling. Okay, so when you release a molecule, a signaling molecule, which acts on your own receptors, that's called autocrine signaling. Okay, and what this then does is sends signal 3 to the T cell, and now the T cell is going to undergo full-on activation. So what's going to happen to this naive CD4 positive T cell once it has received signal 3? Okay, so at the moment we still have a single naive CD4 positive T cell which is still sitting within the lymph node. Okay, so this is our naive CD4 positive T cell, and it's still in either the cortex or the paracortex of the lymph node. However, once it receives signal 3, it's going to firstly differentiate. So the starting process is that it differentiates. It differentiates and matures somewhat. Okay, and the type of cell that it differentiates into is what's known as a T helper naught cell. So this is called a T helper and then naught cell. Okay, so this first process that you're going to undergo is differentiation from a naive CD4 positive T cell into a T helper naught cell. Then what's going to happen is the T helper naught cell is going to proliferate, and you're going to get a whole population of T helper naught cells. Now I will just draw 3, but you're going to get thousands, if not millions, of these things. Okay, so you're going to get an entire population of identical T helper naught cells, and these are all still in the lymph node. Okay, so you've got all of these being produced in the lymph node. Okay, so this next process is proliferation. Okay, right. So you've produced all of these T helper naught cells which now have this identical T cell receptor which is armed against the allergen fragment. Now, the next process that's going to occur is a process that isn't well understood, okay? Because the pathogen was extracellular, okay, you're going to these T helper naught cells are going to differentiate into T helper 2 cells. Okay, so most of them, if not all of them, well, it won't ever be all of them in reality, but as far as our simple little model is concerned, they're going to differentiate into T helper 2 cells. Okay, and the driving force for this differentiation is not understood. However, what is known is that interleukin-4 seems to be a very powerful molecule in driving T helper naught cells to differentiate into T helper 2 cells. And it seems to signal that the pathogen is extracellular and therefore needs a humoral adaptive immune response against it. So humoral just means, well, of the fluids of the body. So it means it's within the interstitial fluid, i.e. it's not within the cells themselves. Instead, it's within the extracellular fluid. Okay, but how the fact that it is an extracellular pathogen is translated into a rise in interleukin-4 is not understood. We don't know what produces this interleukin-4 that triggers the T helper naught cells to differentiate into T helper 2 cells, but something does. Okay, now these T helper 2 cells all still have this T cell receptor which is armed against this allergen fragment, and they are going to help with the activation of B cells. Okay, so we're going to abandon our talk of T cells for now, and we're going to turn our attention to B cells temporarily. Okay, so back to B cells then. So, B cells then. Right, so let's draw a B cell here. So, again, you have a whole bunch of B cells within the lymph nodes, and they are sitting within the B cell follicles within the cortex of the lymph nodes. Now, B cells are not as complicated as T cells, okay? So you don't have to worry about, CD4 positive, CD8 positive. There's nothing like that for B cells. A B cell is a B cell, so we have loads and loads of B cells. However, again, they have a different receptor that recognizes a different antigen, basically. So no two B cells will have the same B cell receptor. Okay, so let me draw a B cell receptor here. So, B cell receptors are effectively a modified antibody that is modified so that it can be implanted into the phospholipid bilayer on the surface of the B cell. Okay, so this B cell could produce the soluble form of this B cell receptor, which would be its antibody. Okay, so it's the same gene that it's using, it's just splicing it slightly differently. Okay, so this is the B cell receptor. Okay, and the B cell receptor is often abbreviated to BCR for short. So, in the B cell follicles within a lymph node, you will have a humongous great number of B cells, okay, and they all have different B cell receptors. Now, what is going to happen? Well, some of the allergen has gone through this antigen-presenting cell process where it's activated T cells, okay? However, some of that allergen, remember, got into the lamina propria. Okay, so we inhaled the allergen for the first time. Some of it got into the lamina propria, where some of it was then phagocytosed by professional antigen-presenting cells, which led to the production of these T helper 2 cells. However, some of that allergen will go straight into the lymph vessels, which lead to this lymph node. So some of it will be coming to the lymph node, and it will be percolating through the cortex and then the paracortex of the lymph node. So it will be in contact with the B cells. Now, maybe there is a B cell that, well, if you are going to launch a humoral adaptive immune response against it, there will be a B cell somewhere that had a B cell receptor which would bind to the allergen, the intact allergen now. So it's not an allergen fragment, this is the intact full allergen here. Okay, so here is our allergen. And again, this is a spot in this pathway where allergic individuals could differ from non-allergic individuals. Potentially, non-allergic individuals do not have B cells which have B cell receptors which will bind to the allergen, okay? And allergic individuals must do. Okay, so here is the allergen. It's now bound to the antigen binding region of the B cell receptor. Okay, so we've searched long and hard, found a B cell that has a complementary B cell receptor to the allergen. Now what's going to happen? Well, we're going to have to draw a big picture for this. Okay, so what's going to happen is the B cell is going to phagocytose, or at least internalize, that entire receptor. Okay, it's not going to phagocytose it, forget I said that, it's going to endocytose it. Receptor-mediated endocytosis is what's going to happen. So, you're going to get an endocytic vesicle, and I realise this endocytic vesicle is drawn absolutely enormously. Of course, it's completely out of scale, but in order to show the B cell receptor properly, I'm going to have to draw it like that. Okay, so here is our B cell receptor. Okay, here is the allergen bound to the B cell receptor, so we'll colour that in blue. Now, B cells are another professional antigen presenting cell. Okay, so what's it going to do? Well, it's going to chop up that allergen into fragments, and it's going to stick those fragments on which protein complex? Well, whenever you get a protein from the extracellular fluid and you endocytose it and then you chop it up, you put the fragments on MHC class 2. That's a general rule. Okay, so here is the MHC class 2, and it's just going to happen that the way that this B cell chops up this allergen fragment is going to mean that we get the same allergen fragment that we launched to this entire T cell response again. So these T helper 2 cells are primed against this very allergen fragment that is now on the surface of these B cells presented in MHC class 2. Okay, so I'll colour in the MHC class 2 in red here. In fact, I might outline. Okay, so outlined in red here is the MHC class 2. Now, the B cells also have to put co-stimulatory molecules on their surface. If they want to get T cell help, they have to put co-stimulatory molecules on their surface. Okay, so again, something must activate the pattern recognition receptors on the surface of the B cell that then leads it to express these co-stimulatory molecules. So it expresses again CD40 here, and either B7.1 or B7.2. So I prefer the names B7.1 or B7.2 as opposed to CD80 and CD86. Okay, so I'll use those because everything's called CD. So it's nicer just to have one little variant on that and use B7.1 and B7.2. Okay, so in green is B7.1 or B7.2. In orange here is CD40. Okay, now this B cell is now ready to get T cell help, and again, this is a point. This putting on your surface of co-stimulatory molecules, again, this is a mystery. What triggers that? And whether that is the process that is different in allergic individuals and non-allergic individuals is still controversial. Okay, so what is going to happen, however, in an allergic individual is that you will get the allergen fragment presented on MHC class 2, and the B cells will somehow end up with co-stimulatory molecules on their surface. Okay, now this B cell has to then find a T helper 2 cell. Now this isn't too difficult because, remember, we're still in the lymph node, and the lymph node now has an absolutely massive great population of these T helper 2 cells that all have a T cell receptor which is complementary to this allergen fragment. Okay, so even though there's only one B cell, it doesn't take too long to find a T helper 2 cell that's geared against the allergen fragment which it has presented on its surface in MHC class 2. Okay, so here then is what's going to happen. The T helper 2 cell here is going to bind its T cell receptor to the MHC class 2. So let's colour in the T cell receptor in purple here, vivid purple. Okay, and then what's also going to happen is the T helper 2 cell will bind its CD4 molecule onto the MHC class 2 on the surface of the B cell. So here is the CD4 molecule, which I will draw in blue here. So this is CD4, and let me just label these up. Okay, so this is CD4. Okay, and then also you have the T cell receptor here, TCR. And then you also have the receptors for these co-stimulatory molecules here. Okay, so for B7.1 or B7.2, this receptor is CD28, and for CD40, it's CD40 ligand. Okay, right, so we form one of these immune synapses, as it's called, and I didn't use that word previously, but it's a very good word to use. So this connection between two immune cells involving the MHC class 2, the co-stimulatory molecules, and their receptors on the surfaces of the T cell, this is called an immune synapse. Okay, and we'll use yellow for CD28. Okay, right. So, what is the result of forming this immune synapse here? Well, what's going to happen is that you're going to end up activating the B cell. Okay? So, firstly, the molecules themselves, the CD40, the B7.1 or B7.2, and the MHC class 2, binding to these molecules on the surface of T helper 2 is going to send signals which activate the B cell. In addition, the T helper 2 cell will send molecules that activate the B cell. And this overall then is known as T cell help. Whoops. Okay, so what's going to happen is the B cell is going to get T cell help. Now you might think, well, surely what happens now is the B cell just divides and divides and divides and divides, differentiates then into plasma cells, and then those plasma cells go out into the blood and produce a huge amount of antibody. Now, it's slightly more complicated than that. What happens in the lymph nodes is really quite clever, really subtle, and this is the most difficult bit to understand of the entire humoral adaptive immune response. Okay, so that doesn't happen, not yet. Instead, what happens is the B cell does divide and divide and divide, but the B cells aren't going to differentiate into plasma cells yet, and they're certainly not going to leave the lymph node. So, the B cell is going to be activated to divide and divide and divide. You're going to get a whole population of B cells. Okay, so here are these B cells. And this will trigger what's known as a germinal center within the B cell follicle. So remember, we saw these B cell follicles over here within the cortex of the lymph node. Well, once one B cell in the follicle has decided to divide and divide and divide and divide, you get a little distinguishable population within the B cell follicle. So if this is the follicle, what you will see is a little subpopulation like this, which is all the cells which have been produced by a single activated B cell. Okay, and this is what's known as a germinal center. So basically, these cells in the germinal center, they are histologically separable from the naive B cells, the B cells that were just sitting inactive. Okay, so they look different. So you can easily see, you can easily distinguish a germinal center from the naive B cells. Okay, so it's histologically distinguishable. Right, now, these B cells that are in the germinal center are not all going to be identical. Okay, so they're all going to have slightly different B cell receptors, and you might say, what? But they were all produced from a single B cell. How on earth do they all have different B cell receptors? Well, basically, what's going to happen is a process known as somatic hypermutation. Okay, so you're going to introduce tiny little mutations into the portion of the B cell receptor which binds to the allergen, basically, this portion up here. So all of them are going to end up with slightly different B cell receptors. Now, why are we doing this? Well, basically, we want to initiate a process of selection. We want to improve the B cell receptor. We want to make the B cell receptor even better than it is now. We want to make it so that it binds to that allergen ridiculously well. We want to increase the affinity of the interaction between the allergen and the B cell receptor. Now, how do we do that? Well, we could start a process of natural selection. Okay, so how do you start a process of natural selection? Well, firstly, the starting point is that you need variation within the population. Okay, so you need a whole population of cells that have differing affinities for binding to the allergen. Okay, so a whole population of cells which have different B cell receptors which will have differing affinities for binding to the B cell receptor, for binding to the allergen. And then what you need to do is select for those B cells which then have B cell receptors which have the highest affinity interaction with the allergen. Okay, so only those ones can survive and the other ones must die. Okay, and then if you continue that process, then eventually you'll end up with a population of B cells that all have very good B cell receptors as far as binding to the allergen is concerned. So let me explain this in a bit more detail. So, all of these B cells that we've produced have got a slightly different B cell receptor. Okay, now I've drawn 3. In reality, you will produce thousands, if not more than that, millions. Okay, so let's call this B cell receptor 1, B cell 2, and B cell 3. And let's say B cell 1 underwent a mutation that actually meant that its B cell receptor was slightly less good at binding to the allergen than the original B cell receptor of our original B cell. Let's say B cell 2 underwent a mutation that meant that its B cell receptor was slightly better at binding to the allergen than the original B cell. But let's say B cell 3 undergoes a fantastic mutation that means that its B cell receptor is just fantastic at binding to the allergen. Okay, now what's going to happen is that within the B cell follicles there are a certain type of cell, okay, known as a follicular dendritic cell, And again, these have dendrites, basically. Now, they have no relation, again, to neurons, and they are very different from the dendritic cells that you have that were involved in the professional antigen presentation. This is a follicular dendritic cell. What they share is that they all have these dendritic processes. So this is a follicular dendritic cell. Okay, now, the purpose of these follicular dendritic cells, which by the way are often abbreviated to FDCs, is that they sit in the B cell follicles, and as allergen percolates through the B cell follicles, trying to get from the subcapsular sinus to the medullary sinus, these will grab onto it. Okay, now they won't just do this for the allergen, they do it for all sorts of proteins that move through the lymph node. Okay, so they'll end up with allergen bound to their surface. Okay, so this means that the B cells are always continuously exposed to allergen, basically, or antigen. And more generally, this will be all sorts of antigens, basically. Now, basically, this means that our B cells are now capable of, they've got allergen supply. They don't have to sit and wait for allergen to be delivered from the lymph, basically. Instead, they can just go to these follicular dendritic cells and try and get the allergen off the follicular dendritic cells. Okay, now, B cell 3 has a fantastic B cell receptor for binding to the allergen, whereas B cell 1 and 2, yes, okay, B cell 2 had an increased affinity B cell receptor for the allergen, okay, but it can't compete against B cell 3. So who is going to get the allergen, basically? It's going to be B cell 3. So imagine there's only 2 pieces of allergen rather than 3. B cell 3 will bind both of these pieces of allergen to it, and there will be no allergen left for 1 or 2. Now, basically, B cells which get allergen will survive, and B cells which don't get allergen will not survive. So this is how we set up our selection pressure in favour of B cells which have a B cell receptor with a higher affinity for the allergen. Those that bind allergen will survive and reproduce. Those that don't will die. Okay, so gradually that is a nice selection pressure in favour of B cells which have a high B cell receptor affinity for the allergen. Okay, so what happens to B cell 3? Well, it's going to bind the allergen to its antigen binding region of the B cell receptor. And then what's going to happen is the exact same process that happened to the original B cell. Okay? So, it's going to endocytose the whole B cell receptor with the allergen bound to it. It will then chop up the allergen, put fragments on MHC class 2. It'll also get co-stimulatory molecules, where from, again, is a bit of a mystery. And it will then interact with T helper 2 cells and get T cell help again. And then what will happen? The B cell 3, will become activated, and it will divide and divide and divide again. And again, the B cells will undergo somatic hypermutation. So although all of the offspring are based on the original B cell, and their B cell receptors will be based on the original B cell receptor, they will all slightly differ. So basically, we're then going to make another population, all of which are based on B cell 3. Okay, and whereas B cell 1 and 2, when they don't get allergen, they won't be able to present the allergen fragment to T helper 2 cells. They won't get T helper help, okay, and they won't survive. So you don't survive unless you present the allergen fragment to the T helper 2 cells. The T helper 2 cells forming this immune synapse with you gives you the signal to survive. It gives you more than that. It gives you the signal to survive and then divide and divide and divide. So you can see that over time what's going to happen is that you are favouring the B cells which have high affinity B cell receptors for the allergen. Okay, and those B cells are surviving and reproducing, whereas the other B cells which have lower affinity B cell receptors for the allergen, those are dying. Okay, so over time, what's going to happen to this population of B cells within the germinal center? Well, it's going to gradually evolve so that now have a population of B cells, all of which have B cell receptors which have a very high affinity for binding to the allergen. Okay, so overall, gradually in this germinal center, the population will evolve, and you'll get a population where all the B cells have extremely high B cell receptors, have extremely high affinity B cell receptors for binding to this allergen. Okay, and we'll continue this discussion in the next video.